It's my great pleasure to invite Professor Naftali Kaminsky to give the Margaret Turner Warwick Centre for Fibrosing Lung Disease seminar. I've known Naftali for many, many years. We did postdoctoral fellowships together. Well, you're a little bit ahead of me, but in the same lab. I followed in his footsteps in Dean Shepherd's lab over 20 years ago now. And since that time, Naftali has gone from strength to strength and is the world leader in omic research in pulmonary fibrosis. There's no doubt about it. So I'm going to keep the introduction very short because we've already wasted too much time with the technology. So thank you, Naftali. It's a great honor to have you here, and I look forward to hearing all about your work.
Thanks for the invite. It's a pleasure to be here. It's really exciting. And now that Rachel has entered, we can start. Yeah, we just waited. Good. So again, thanks, and really a pleasure to see you. I'll tell you a few stories here about our single cell work and how we think about pulmonary fibrosis now, and trying to incorporate potentially some bioengineering principles to our understanding of the disease. These are sort of my conflict of interest, and I'll start with a little bit touching on fibrosis, principles of tissue organization, cellular organization of the lung, what happens to lung cells in pulmonary fibrosis, and try to present the model. So fibrosis is actually not a very old world, and it was sort of comes from, fiber entrails or something like this, and was not really introduced into human disease until relatively long and late. And basically, fibrosis is the replacement of normal parenchymal tissue with connective fibrous stuff that impacts the function of an organ. And as a reminder, an organ is a multicellular of structure of the organism that carried out a specific function. We tend to forget about it. We tend to focus about one cell or the other. And fibrosis is an example of disease that is actually not a disease of the cell, but a disease of the organ inheriting its function. If we look at fibrotic organ diseases, you all know this: heart, liver, lung, gut, kidney, skin. They all have fibrosis. There's some commonalities. I think the most common commonalities, one is exposures. The second is overload, when the tissues are overloaded. The third is autoimmunity, which is sort of a common thread about all of these. When we look at the pathways, this is just a generic pathways of all the pathways associated with fibrosis. I'm not going to go all of them. Interestingly, they're found in every organ. So if we look at the papers, actually, some of the papers look very much like copies of each other. So the pathways are similar. However, as you all know, there's a minimal genetic overlap, and actually almost no genetic overlap between the different fibrotic conditions, except for actually telomerase pathways genes. These rare mutations, or sometimes common mutations, will show up in almost every one, but all the others are usually organ-specific or overload-specific. The other thing, this is substantial cellular overlap. Most people will speak about fibrosis in the context of fibroblasts. That's not what I'm referring to, and I'll be more clear later. So, of course, the disease I'm most interested in is pulmonary fibrosis. There's sort of two groups of diseases. One is idiopathic pulmonary fibrosis. The other one is progressive pulmonary fibrosis. Around 500,000 people in the US alone. Only cure is lung transplant. They may be caused by genetic, environmental exposure, or autoimmune idiopathic. The most important thing is it's most lethal when it's associated with this pattern, which I find mostly fascinating. And since I looked at IPF lungs around 30 years ago the first time, and probably more seriously around 20 years ago with Sam Houston, this sort of structure, the disease that comes from the periphery, is relatively spread equally in the lung, is characterized by honeycombing and traction bronchiectasis, and in the lung, the interspersing of normal lung and disease lung with the sort of the fibroblast foci, with sort of the can you see this, or with a sort of the fibroblast foci, the layer of epithelial cell around it, a little bit of vascularization, and these lymphoid cells that are sort of always not in the cutting edge, but are so always hovering around. All of this is sort of very typical. And the paradigms of idiopathic pulmonary fibrosis, I think all of you are familiar. I think the biggest changes over the years has been basically the fact that the disease have several rates of progression. The disease can be stable, can be progressive, or can be sort of shifting between. The fact that there's no evidence of real sort of old-fashioned traditional inflammation, the kind of inflammation that I was trained in, role for fibroblasts, epithelial injury, active remodeling, a role for aging, exposure, re-hour stress, mitochondrial dysfunction. All of these have been introduced in the last decade. Strong genetic component, 30 to 50% of the patients are associated with genetic aberrations, most common MAC5B. And then there's the rare survival, 3-5 years from diagnosis, hasn't been changed much. Immunosuppression kills our patients. Now, I will draw here a small distinction. Immunosuppression, not immunomodulation. And I think that's a point that we need to think about later. And some drugs can slow down the disease. What is our working hypothesis about the disease? Basically, this is the typical lesion of the disease, and many of you have seen me show these GIFs, just GIFs that I made probably 9 years ago now. Basically, reiterating Moises Selman's original hypothesis paper, which you have recurring microinjury that hurt epithelial cells, cause epithelial cell injury, epithelial fibroblasts and macrophage activation, matrix remodeling, cell proliferation, migration. If you're genetic predisposition, exposure, telomere attrition, you're going to develop fibrosis. So the truth is that this model is really cool and sounds good, but it actually does not explain much because it doesn't tell us what is the cellular repertoire of the lung. And when you look at the tissue, you can tell there's multiple cells involved. It doesn't tell us the anatomic features. Why do you get these foci? What is peripheral? It doesn't explain the relentless progression. And all the people calling, like, even I sometimes tell patients, okay, that's scarring of the lung of unknown reason. The truth is that scars don't progress, right? They contract. And actually, if you look at CT scans of kids that have had radiation for lymphomas, it's very rare that they have fibrosis, this sort of line of fibrosis that does not respect anatomical barriers that you see in radiation, almost never progresses. If anything, it shrinks. And you all know this, you all have scars. They rarely progress. So I think there's a very interesting question there. And then, of course, I use my cat's brain sometimes. This is actually not my cat, but it's actually my inner cat that's thinking. And the answer is no. If we think about it in an unbiased way, this model doesn't really explain the disease. So just reminding people about the sort of the basic principles of tissue organization in the transition from a unicellular organism to a multicellular organism to a multi-organ organism. Tissues like humans cells like humans had to agree to be in a community and expect certain rules. And every tissue has a primary cell that carries the function of the tissue. That's a really universal principle. Every tissue has support cells that has the primary cell carry its function. You can have cells that sense, feed, maintain, support, clean up, a lot of it. But the roles are not constant. So it's not like an epithelial cell is always going to be the primary cell or endothelial cell. In every tissue, it's going to be a little bit different. And a cell that is a support system in one organ can be a primary cell in another tissue. And also the tissue itself can be a support system. There's a huge discussion in this, and I follow Ruslan Majidov that always says, I think it's Ruslan, maybe somebody else has said, we know that if we know one thing for both science and human history is the brain is not the organ that we exist for. Probably the reproductive system, like every other organism in the world, but that's not, as a pulmonologist, that's not what I go into. But what I want to remind you all is this hierarchy. There's a cell that does the function of the tissue. There's cells that do support system. And this is just examples. I won't go into this. If we're thinking about the lung, in this terms of analysis, this is a very simple cartoon of the lung. You have the airway, moving cells, moving air in and out, cleaning up, making sure it definitely is a support thing. And then you have the alveolus. This is what the lung exists for. And in the alveolus, it's basically the alveolar type 1 and potentially the aerocyte, which is why the lung exists. The lung exists for gas exchange. And the alveolar type 1, this high surface cell that does actually do so much, is probably the most important cell. And then there's a lot of support cells in the alveolus, the alveolar macrophage, which probably is the sensor cell, the alveolar fibroblast that is there mostly because it was there when the lung was built. But also it's supposed to do some things when things go wrong. The general capillary cell, which is feeding probably the aerocyte, we don't know yet. And of course, the alveolar type 2 cell. And the alveolar type 2 cell does a lot of things. And when you look at this, you understand something, and that's why I like this simplified image. You basically understand the structural constraints of the lung. One of the reasons that the lung is so effective and efficient is because you need to move gases across a very thin barrier. So you cannot have many cells doing many things, and every cell in this area is doing several functions. So to understand the cellular structure of the lung, again, around we started doing single cell analysis of human pulmonary fibrosis lungs, probably around 6 years ago. And then in 2019, during the pandemic, we released, during the 4th month of the pandemic, we released the IPF Cell Atlas, which was basically a database of all the single cell data, up to date. It's a big success. We've had 15,000 unique users from 56 countries and 5 continents since inception. A lot of queries. This is the collaborative work. Taylor Adams and Jonas Schupp and Nir Neumark, who sort of started it with the Zitin Young and Farida Angari. It was a really good collaboration with Ivan Rosas, who actually created the tissue resource. And the website was actually created by 2 college graduates, Carlos Cosme, and later continued by Jasper Flint. Both of them are now MD PhD students. And it has both the data of other groups and our groups. And if you haven't used it, feel free to look at it and use it. I think it's really helpful. We'll soon release the IPF Cell Atlas too. So what did we learn from the IPF Cell Atlas? What happens to the primary cells of the lung? So as I told you, and there was no debate about it, at least in this room, the ATD1s are probably the most important cells in the lung, or the most primary functional cells in the lungs with the aerocytes. And when you look at the IPF Cell Atlas, it's very easy. And what you're seeing here is basically blue is control, yellow is COPD, red is IPF. And in explant lung, these are end-stage lungs. Basically, there's a clear loss of alveolar type 2 cells. If you look at cryobiopsies, these are biopsies of patients that have interstitial lung anomalies suspected as IPF, but diagnosed with IPF after the biopsy, and when follow-up, this was proven. Basically, we see the same thing. This is control. Yellow is early IPF. Blue is late IPF, and this is other interstitial lung disease in this patients who are very asymptomatic. I will be honest, most of them have FVC, the forced vital capacity that's what's called normal, above 80%, but their DLCOs are below 80%. So most of them already have disease, even though they're asymptomatic. And basically, you can see a decline of 81s early in the disease, and also a decline in 82s. What about aerocytes? The same thing. Again, this is G cup and A cup. Blue is control, yellow CPD, red is IPF, and you see this decline, and it happens also in early disease. Sorry, it's here. So here are the G cups, and here is the A cups. So basically, IPF is a disease of loss of the gas exchange unit. What happens to key support cells in the lung? Again, 82s are basically lost, and this is when you look at the 82s, I've already showed you this at 82 loss. If we take samples and grade them based on human sample based on the disease involvement or progression of fibrosis, basically you can see there's a gradual loss of 82. Blue is 80, AT2, and green is AT1. So basically, they're lost as the disease progresses. What about endothelial cells? As I said, AT1s are lost, erythrocytes are lost, but also general capillary loss. And what drew our attention a lot is that actually in the alveolus, we saw an emergence of it. Endothelial cell population, we could not really classify. This Jonas Schuck was sort of overcoming my resistance to look at the endothelium and forced me to look at this data. And basically, we saw a unique population of cells that express collagen 15, they express venous endothelial markers, but not fully perfect. And when we stain for collagen 15, we never see these cells in control lungs. We do see them around bronchial vessels, and here's around central vessels. And we see them also in the pleura, but in IPF, we see these positive cells usually on these areas of neoangiogenesis. Sometimes they're captured, but that's like the other side of the fibroblast foci. And Jonas has published the endothelial cell atlas in Circulation. You could look for details. This is a really interesting point, and this also reminded me of something. Some of you heard me tell the stories when I was a little kid. 4 or 5, I thought I was really smart. So every day I had a new idea, and I would go and tell my grandfather my idea. And my grandfather would say, well, you need to learn to read. Because if you learn to read, you can see that all your new ideas were written by people before. Not the most supportive grandfather, but really made me competitive. Was actually. And so if we look at this phenomenon, actually, it's not a novel phenomenon. It's a pleasure to remind the people here on this opportunity, actually, which is the paper by Margaret Turner Warwick from 1963, in which she describes sort of precapillary systemic pulmonary anastomosis and describes this phenomena in IPF lungs. They still call the disease systemic pulmonary fibrosis, but only in the text of they say that it's equal to interstitial fibrosis. I think they interpreted some of these anastomosis as arterial anastomosis, but again, the fact that these anastomosis we all sort of trained our eyes to ignore, or these neoangiogenic anastomosis sort of show up and stare at us in single cell data suggests that we study this. This also highlights this idea of loss of the gastric change unit and potentially also increase in shunt in the IPF lungs. And of course, once you start looking for stuff, it shows up. In recent years, there have been several papers. Jeff Whitson's group has shown that in alveolar capillary dysplasia, a disease in which, congenital disease in which there is a mutation in FOXF1, erythrocytes don't develop. General capillary cells do develop, alveolar type 2 cells develop, but alveolar type 1s don't develop. So it's a failure of the gastric change unit. And in these lungs, also there's a lot of collagen 15 endothelial cells, and then more recently it was shown also in NSIP, leading Jonas to feel very excited about it. The idea that 4 years ago we did not know that they existed, and now we find them everywhere. And yes, maybe that's true. Once we make a distinction, we tend to see it. But it's also a warning sign, because once you publish something and it's novel and interesting and exciting, people tend to find it in other places. And if you focus on one marker, you may actually misidentify. So again, and you'll hear me say it again and again and again, important to look not at the single cell marker, but actually focus at the gestalt of the cell. What about fibroblasts? So everybody's been thinking about fibroblasts, and we learned a lot about myofibroblasts. The truth is that the cells we called myofibroblasts in our initial analysis didn't have, many of them didn't have alpha-smooth lactin. And as I sort of look at the data and look now at very more data sets, it's actually the key cell that is profibrotic or associated with fibrosis is a cell that is enriched with the molecule called THRC1. This was described by Dean Shepard, both in the collagen, the Atlas of Collagen Producing Cells a few years ago, and then more recently the paper in Nature. And they're definitely representing sort of the cutting edge of fibrosis. If you look at their data here, this is all mesenchymal cells, red is IPF, blue is control, and you have this thing that never shows up in controls. And in this region, you have CTHRC1 cells, and they express a lot of other what I call Betty molecules like periostin and NOX4 and twist and ADAM TS19, which is probably sort of the cutting edge of the fibrosis. But of course, there's more characteristic. And I think when we looked at the data, I don't think I'm actually know that we looked at the data. One of the things that came to our mind is we cannot really define in the fibroblast populations real subtypes. We see cells that are characterized by what I call functional characteristics. And this is work actually done by Miri Adler when she was in Ruslan's lab. And what they did is they took our fibroblast data and instead of clustering them, they looked at their disbursement across functional ideas. And what they did, they implemented an economics model, which is the Pareto model of use of constricted resources. And the idea in the Pareto is very simple. If you live in a big city, you're going to have a person that's selling shoes and a person that's selling shirts and a firefighter and a policeman and a teacher. If you live in a village, the barber is also going to be your doctor. And if there's a fire, everybody becomes a firefighter and on and on and on. So the idea about the lung, the fibroblasts, is that they have this plastic ability. And then you look at normal fibroblasts, they're just generalists. They have all these capacities. And in the IPF lung, despite the fact that they don't change, you have sort of populations that do contractile fibroblasts, protein biosynthesis, extracellular production. But this is actually interchangeable. What about macrophages? There's been a lot of discussion about macrophages in IPF, so I won't go into this, but I'll mention that in every single cell data set, you find these sort of SPP1 positive fibrosis-associated monocyte-derived alveolar macrophages. This is sort of the cluster of these cells. Again, red is IPF. This is a list of their markers. YKL40, MMP9, osteopontin, PLA2G7, which actually is an enzyme that can make LPA, which is sort of an interesting. There's a discussion why two trials that inhibit endotoxin fail. There's actually a lot of enzymes that can make LPA in the human lung, MCP1, TIMP1, and on and on. Amy Zhao in my lab integrated the peripheral blood mononuclear cell data, BL, and lung cell data, and did pseudotime analysis and other things, and showed that basically SPP1 where it did monocyte or suggested monocyte-derived alveolar macrophages. And of course, when we looked at the data initially, the one clear thing that we found was that there were epithelial cells that we could not classify. We ended up calling them aberrant basaloid cells. Actually, when we submitted the paper, we called them epithelial alveolar mystery cells, but reviewers suggested that we omit. I refused to name them based on markers. Actually, at the time, I already had discussion with John Krupski, who we knew had a similar marker. So he decided to go with the markers, and we decided to go with the concept. And the reason I didn't want to use the markers is because these cells were very unique because they had a very complex set of markers, including what we call basaloids. They express P63, keratin 17, beta-lac keratin 5. Usually airway basal cells have P63, keratin 17, and keratin 5. They actually lose another really important regulator of, which is in airway basal cells, which is MIR205 host gene. They carry EMT markers. They have fibronectin, collagen 1A1, and N-cadherin. Actually, they're the only epithelial cell in the lung that has N-cadherin. They have the machinery to activate TGF-beta. They express the integrin alpha beta 6, which both Gisley and I studied in the Dean of Shepherd, in the lab of Dean Shepherd, and now is studied as a molecular target for IPF. They have machinery to respond to lipophosphatidic acid. Including, again, the LPA receptor 1, but also an alternative receptor called GPR87, suggesting, again, that the interventions we're trying in LPA probably not targeting everything. They express the most P21 in the human lung. Actually, if we look at it, they're the only cells that really light up with P21, suggesting that they're senescent. They also express a lot of GDF. 15, they carry stemness signatures. And they also express MMP7, the same way like every basal cells in the lung. MMP7 is sort of a IPF marker that I stumbled upon at the end of my postdoc, and actually published in PNAS saying that it was expressed in alveolar type 2. Probably what I saw there were aberrant basal cells, and has been found since then in the peripheral blood, including a meta-analysis. So it's probably the most validated outcome marker in IPF. So TGF-beta activation, LPA response, and SMCMT, MMP7 B cells are the Swiss Army knife of fibrosis. I really got a kick of it because this year I actually got to say this in Switzerland, which was really fun. But the same sort of my grandfather's paradigm. When you find something new, look at the literature, it's probably there. So I think they were probably first observed in 2002 by Marco Chilosi and Venerina Politi. They published that basically alveolar cells at the edge of fibroblast foci, maybe type 2 cells, but they expressed the exaggerated Wnt program, MMP7, and potentially other things. And then Zia Boruk in 2007 published a paper about epithelial mesenchymal transition in IPF. That paper was highly doubted because of data in mice that suggested that full epithelial mesenchymal transition going from epithelial to fibroblasts is impossible. However, the original observation in the human stains is actually confirmed by our data. These cells carry mesenchymal markers, you just don't become fibroblasts. Also, we've analyzed almost every single pulmonary fibrosis paper, regardless whether the authors noticed it or not, and found that they're also found in end-stage fibrotic COVID-19 lungs. They found in spatial transcriptomic analysis of IPF lung, both in the data from Meyer and from Franzen, recently published. They're rarely observed in COPD, never normal lung. But the thing I want to highlight again, they're characterized by a complex functional gestalt. This is not a combination of two markers. You want to think about as the combination of senescence, EMT, stemness, and IPF markers, probably better, and basalness, which we define by the expression of P63. Where do aberrant basal cells come from? So the truth is, I don't really know. But when we thought about it, we thought that one way to try to see where they come from is basically to analyze differentially affected regions in the IPF lung. If you look at an IPF lung, you will find areas that are sort of in the relatively unaffected end stage or at the edge, and they have this sort of histological characteristic. And there's a resource that's created based on initially by Jim Hogg. Jim Hogg, world-renowned pathologist, created an algorithm in which we do a method in which you take explanted lungs, you freeze them over liquid nitrogen, and then you do a CT scan out of the body. This CT scan is actually high resolution because you don't have a motion artifact. You can actually nuke the lung as much as you want. Then you can cut the lung into slices. You pick areas that are less affected, non-affected. You can understand the structure. You could get these cores. And this is how a control lung looks. We can actually do a micro CT in early IPF, sort of a middle involvement IPF and end stage IPF. This is run by Bart and Wim Voits at Leuven. And this is Wim standing pounded, part of his freezers that have 718 lungs. It's an impressive proportion. And we had a really great team that analyzed the data, including Jim McDonough, Steve Bar-Joseph, June Ding, Farid Angari, Jonas Schupp, Taylor Adams, Sitini Young, and most recently, Nibala Hussain. And we actually took advantage of this resource long ago. We did bulk RNA-seq and identified differentially transcriptional trajectories and actually used this to develop computational tools for regulatory networks long ago. So what we did is we take these lungs and we took 12 IPF, 11 controls lungs, and we analyzed 3 regions in every one of these lungs. And the first thing, and we did nuclear seq and not dissociated cells. So this was the first thing that we did. If you look at all the dissociated cell data sets in IPF, you would think that the lung is actually a granuloma and inflammatory organs. There's around between 70 to 80% of the cells are inflammatory. When you do nuclear-seq, you get a much better and more realistic fraction of the lungs, with 24% of the cells being inflammatory, equal number of endothelial and epithelial cells, and 11% mesenchymal cells. We identified all the known cell types, and we found actually the same features. So that's really reassuring. Another set of samples, different technology, we find loss of alveolar epithelial cells, loss of endothelial capillary cells, fine ciliated cell in the alveoli, collagen 15A1, venous endothelial cells, and the aberrant basozoid cells. The thing that we did not expect was actually this, what I call the deluge of intermediate alveolar epithelial cells. Basically, when we cluster the alveolar epithelial cells, we see the type 1s, the type 2s, and the aberrant basozoid cells, but we see a lot of cells in between. This is sort of how the data looks. This is sort of the clustering with the markers, and again, you can see sort of the decline in 82 and 81s and all of the other features that I mentioned. This is a representation of the data. When a lot of people show UMAPs or T-SNE plots of single cell data, there's no magic in these things. Basically, it's impossible to look at single cell data. You never see it. So what we take, it's a bunch of numbers, we cluster them, we put things that look the same next to each other. It's relatively simple. The software actually doesn't decide on the structure, and you can make this figure into an elephant or into a monkey if you wish. But what you see here, every dot is a single, is a cell, cells that are looked to each other, and we sort of classify them based on our markers. And in our group, it's a mixed iterative process, which means we'll cluster the samples, we'll annotate the cells, we recluster them, we'll reannotate them, and we test each other. And sometimes it'll take a few months, because if you just take off-the-shelf software and cluster the cells, you're going to make errors. So green are, as you see, alveolar type 1 cells are here, and alveolar type 2 cells are here. And here you're sort of secretory cells and basal cells. And here you find the aberrant basal cells. And you see this bridge here, which is going from alveolar type 2 cells to alveolar type 1 cells and going into the aberrant basal cells. And what is cool is, because of this data that we didn't see when we didn't have differentially affected regions in the same lung, you could start to see markers that are going from one side to the other. So for instance, P63 is only in basal cells and in aberrant basal cells, a little bit in type 2 cells, but I don't know that that's some noise. Surfactant C, of course, in type 2 cells. CPA6 is sort of going from type 2 cells to aberrant basal cells to type 1 cells, and cadherin only in the aberrant basal cells, and on and on and on. So this allowed us actually to implement models, and we use basically an iteration of FAGA, and the method is doesn't important is we take the UMAP, we simplify it, we apply a forced directionality, and basically look only at this region, and you see type 2 cells and type 1 cells and aberrant basal cells. And you could see that all the other cell population does not contribute to this, and it looks like alveolar type 2 cells are going into aberrant basal cells or into type 1 cells, and maybe a few type 1 cells are going. I call this the drinking bird model of epithelial plasticity in IPF. And we've run multiple alternatives, and we come constantly with exactly the same result. The other thing, which is more important, this allows us to basically identify markers for early. So there's the loss of surfactant C, the sort of the group of intermediate cells basically express cathepsin A. Then there's a group that expresses CPA6 and GDF15, which increases in the end. Then you have P63 characterizing in the end. And when you do this, you can actually now still do combinations of markers to see if we find these cells. And when we do the stain the lung, we do find combination of cells that express either surfactant C with and without cathepsin C, surfactant C with the CPA6 on and off, and other cells. So this suggests that we are finding these intermediate cell populations in the IPF lung. Can this be modeled experimentally? So if you would ask me, you would have asked me 8 months ago, I would say no. We have tried to isolate aberrant basal cells multiple times and could not. Basically, what happens is when you flow through the lung, these cells, because I think they're senescent, they just sit there and stare at you. and you can never get to enough cells. But luckily, we applied a model, which all of you know, precision cut lung slices. This is a model that you take the lung or a piece of the lung. We prefer lobule because then we can inflate it with low melt agarose. We cut it using a fibromatome, and then we culture these lungs. And people, Melanie Kenigsoff and Darcy Wagner and others, developed the fibrotic cocktail. You put these lungs in the fibrotic cocktail, which is LPA, TGF beta, TNF, I don't remember what else, PDGF, and they develop a little bit of fibrosis. The only thing that most people don't look is actually at the control lung. So Orléans Justet and Johad Khoury in my lab actually did a time course, and they basically harvested the PCLS at 0, 12, 24, 48 hours, 72 hours, 96, and 120. And there were control lungs and fibrotic cocktail. And you can see here that there's cells that show up mostly in the fibrotic cocktail, but a lot of actually there is overlap and there's a time dependent. I won't go into every single data. I'll just tell you one important thing, or maybe two. So the first one is what's called a control is not a control. This is a really sick, miserable lung. And think about it. What did you do? You took the lung, cut it into small slices, and then put it in media. So that's a model for pneumonectomy and drowning. And when you look at single cell, you find very few normal cells in day 0 or day 1. And then you have sort of a dramatic increase in mitosis within the first 2 days. It sort of reaches a plateau. And the cell starts normalizing, and you start seeing a little bit of type 2 cells, and a little bit of type 1s, and a lot of intermediate cells. Now, if you put the fibrotic cocktail, this does not happen. Basically, this bounce of proliferation does not happen. And as many of us from the older generation, maybe me and Rachel and Gisli, I'm not sure about Peter, no, is when you put TGF beta on epithelial cells, they don't proliferate. It's actually well known. You induce P21 and nothing happens, right? So this mitosis, but instead of getting the repair of the lung, you're getting basically the emergence of aberrant basilar cells. And you can see here basically, this is a heat map of all the cell types we identified with the different treatments. So, you know, cocktail, control, different cell types. And basically, and that's this percentage of epithelial cells. And basically, with the cocktail, at day 3, you have a lot of aberrant basilar cells, almost 20% to 25% of the epithelial cells are aberrant basilar cells. Whereas in the untreated, there are a few, but it's minimal. So, and when we looked at the cells, and we look at what we call aberrant basilar cells in the precision cut lung slices, we saw that this is IPF, this is PCLS. We saw that the signature in terms of the cells, we consider the gestalt of these cells very similar. And by the way, there's been some publications about these cells, especially in mice. We almost never find this kind of overlap. But here's another small observation I want to share with you. So remember, I told you that some cells, maybe 7, in the normal unfibrotic cocktail PCLS also express a gestalt of the aberrant basilar cells, but they miss sort of these key factors, the expression of P63, N-cadherin, F8B2, SOX4, PI1, ADAM-TSX, and collagen 1A1, suggesting that there may be sort of cells of the road. And here's an interesting thing. So we now extended our time course. And the truth is that if you wait long enough in the PCLS, you will see the emergence of aberrant basilar cells. It's just not a lot of them. Okay, so how different are these aberrant basilar cells? Are they just the way I told you about fibroblasts? Is this just a functional state of the cell? I'm not sure why I picked this photo of the cat here, but I think it's a good one. It's also reminiscent of my experiences at home. Basically, we have 2 cats. One is really shy. The other one is really assertive. Both of them wake up my wife in the morning because they want food. If I'm alone at home, they don't wake me up, but they'll sit there and stare at me until I wake up like, oh, who's looking at me? So anyway, so that's the idea. So one way to know cell state and cell type is basically to perform ATAC-seq, which is a method that allows you to measure chromatin accessibility. So we took advantage of the introduction of the multi-ohm assay. Basically, we ran ATAC-seq and single nuclear RNA-seq in the same cell, so we get single cell data. This is actually a much harder assay to perform than we thought it would be. Here is sort of the single nuclear RNA-seq UMAP gene expression. So you see the type 2 cells here, the emitter cells, the AT1s, and the aberrant basaloid cells. And you can see also some of the basal cell here and the secretories, but it really replicates the same structure. If we do the attack, the nice thing is this is chromatin accessibility. These are pics, not gene expression. And you can see the data is the same structure with the same cells, or with the bridge cells, the aberrant basaloid cells, the 82, the 81s, and so on. So really reassuring that we see this. And this is sort of the markers, and you could see, for instance, the markers for every cell type, like conciliated or secretory. This is the gene expression. This is the chromatin accessibility. And if you see the aberrant basaloid cells, there's a dramatic opening of these markers that I mentioned, including FRNBT and HMGA2 and N-cadherin and on and on and on. When we look at the sites, this is really dramatically different. So basically, there's a dramatic opening of FOS and June and BAC and sites. And when we look at the commonality, they all share the AP1 motif. And indeed, when we look at the AP1 motifs in all sites in the lungs using chromatin accessibility, you see that the cell types have slightly different peaks. But look at the aberrant basaloid cells, comparing them to basal cells, comparing them to type 2 cells, type 1 cells, and then intermediates. Basically, this is a dramatically different profile. And what's interesting, when we compared it and look into literature, the most associated dramatically open AP1 chromatin accessibility sites is associated with senescence. So clearly suggesting that aberrant basaloid cells are very different than all other lung cells and probably are characterized by a senescence program. But, you know, maybe this phenomenon is a late phenomenon. Maybe just because this is end-stage lungs. So in collaboration with Venerino Politi and Sara Tomasetti, I'm not sure why this thing here is not showing up. That's actually the spatial transcriptomics of the IPF lung, I think. It's not showing here, but I apologize. In collaboration with Claudia Ravaglia, Sara Tomasetti, and Venerino Politi, we take cryobiopsies of these pre-symptomatic patients. They still don't have IPF. And this is sort of how it looks. It's really, you take out nothing out of the lung. And Nabala Hussain and early understood did spatial transcriptomics and basically identified aberrant basaloid cells in these lungs. And when they did nuclear-seq, basically they could show, and they compared it to end-stage IPF or other interstitial lung diseases. I showed you this data for the 81s and the 82s. But this is basically the thing in controls. There's no aberrant basaloid cells. In early ILDs, there's an increase in aberrant basaloid cells, which is higher in the end-stage lungs. Now, I wonder if this is true, which is that the alveolar intermediates are actually much higher in the early disease. I don't know that this is true yet, but hopefully we'll be able to confirm it. And the other thing is that the emergence of aberrant basaloid cells is associated with progressive disease. So patients who progressed in the following 2 years, and we're looking at this. So we have 125 samples, of them 50 are IPFs, and we did snRNA-seq spatial. We're also going to do some lung bowel blood. You all know the way. What about inflammation? This is sort of a little bit the dogma of progressive pulmonary fibrosis in IPF. You have autoimmunity, foreign antigen, other exposure to hepatic genetics, and the assumption is that you have a phase of inflammation and a phase of fibrosis, and this phase will respond to anti-inflammatories, and this phase respond to antifibrotics. I think we should think about it more carefully. And from my point of view, the assumption that an organ will be completely destroyed and the innate immune system, the adaptive immune system, the mucosal immunity will not know about it doesn't make sense. Inflammation may not drive the fibrosis initially, but the sensing mechanisms, the information mechanisms have to work. So there's no sense that we have to study inflammation within fibrosis. And so what do we know about inflammation during fibrosis, not preceding fibrosis or causing fibrosis? We know actually a lot. One is we know that immunosuppression kills our patients, so inflammation has to do something. Second, we know that immunosuppression does not reverse fibrosis. Maybe in scleroderma, but not in any of the other disease. I had a long discussion with Philippe Diodet 2 days ago in Paris about does immune modulation and rheumatoid arthritis affect fibrosis or not. I'm not sure we reached the conclusion, but most of this data is actually retrospective, so hard to assess effect. The other thing we know is that in IPF patients, peripheral blood gene expression predicts outcomes, and basically there's a 52 gene signature that predicts outcome. And then when Pulvesh Khatri did deconvolution of the signature, they suggested monocytes, increased monocytes predict survival in IPF. This was published in Licensed Respiratory Medicine and then confirmed by Michael Crowther and Toby Meyer. And then the other thing, which is interesting, and we tend to ignore it, or at least myself, is that many of our patients with IPF tend to show up with enlarged metastatic lymph nodes. And we even do sometimes EBUS biopsies to rule out cancer, but we don't really think about it, or at least I didn't think about it until I saw the paper from Deji Adegosoy and Ann Sperling showing that patients with IPF increased lymph nodes predict increased survival. And we also know from single cell data that inflammatory repertoire of the end-stage fibrotic lung disease is dominated by fibrotic associated osteopontin presenting cells. So definitely inflammation is relevant to the disease. It's just not necessarily the inflammation we thought about. Rami Unterman, when he was in my lab, basically did single cell profiling of peripheral blood in IPF patients. He did a relatively small but extremely well-characterized cohort. And they did single cell and then did a few other things. And basically, well, of course, I was really happy that when we did the single cell, we found all immune cell types known in the lung, in the peripheral blood. And the first thing he showed, and I was really reassured, is the fact he confirmed the fact that monocytes are increased in IPF, stable, and even more increased in progressive, but we could classify them. Now we know that it's only classical monocytes and intermediate, but not non-classical monocytes. This is their markers. There's actually more markers, but that's in the HLA-2 score, S100A8 and S100A12. And I think some of you will recognize these molecules. And when we look at survival, basically high monocytes associated with worse survival. And this is just confirming work that was done before. So really nice when you do an unbiased analysis and you find things that other people have found. More surprising was Rami's analysis of the lymphocytes. Basically, we all know that IPF patients have less lymphocytes. They're not lymphopenic, but the lymphocytes sort of go down with disease progression. But what we didn't know that in patients who are progressive, there's an increase in Tregs, and this was confirmed in an independent cohort by flow cytometry. And basically, patients with high Tregs have worse prognosis. And we find also increased Tregs in the lungs of patients with IPF. And the lungs we have are lungs of patients with transplant, so obviously they progress. So definitely there's an increase in Tregs. And we are looking now at the presence of Tregs in EBUSs of patients with mediastinal lymph nodes to try to start understanding it. Interestingly, these Tregs are actually immunosuppressive, as are some of the monocytes in the peripheral blood in IPF, suggesting a potential immunosuppressive or exhaustive phenotype in these patients. Amy Zhao, an MD, brilliant MD PhD in my lab, who's graduating with her MD this year and graduated for her PhD 2 years ago, analyzed and compared the blood of patients with the fibrotic HP control and IPF. We selected the fibrotic HP only patients with UIP patterns. This is probably one of the largest cohorts in single cell data in humans, or humans with definitely interstitial lung disease. It's also the first cohort that has samples from how would you call it, a low middle income country from Mexico City. So really proud of this cohort, and Amy did an amazing job of running this. And just to basically summarize this, we don't go into detail. The first thing it's confirmed is that in fibrotic HP, we see in the peripheral blood, in terms of monocyte, we see exactly the same thing as we see in IPF, basically increase in monocyte, S112 cathepsin D. There's a modulus. So I call this basically the roasted chicken model of monocyte behavior in fibrosis. So basically, classical monocytes are increasing their FHB like in IPF. And also we did integration of BAL and PBMC data, as I mentioned, and revealed the classical monocytes that have developed into phosphopontin high monocyte-derived lung macrophages. What happened to lymphocyte? I don't know what happened to my animation, but I'll just summarize the data. Basically, when you look at the T cell lymphocyte, it was very obvious. FHP lungs had just a lot of cytotoxic T cells expressing granzyme A and B, and we also saw as far as granzyme A and B CD4 cells and CD8 cells, but we also saw a unique phenotype in CD8 T cells. We saw granzyme H high phenotype. Granzyme H has been associated with autoimmune disease, and it's been studied also in transplant, so really interesting. And then in the Mexico samples, we found also granzyme K, suggesting some kind of a local terroir, sort of a type of phenotype. So single cell analysis highly feasible for detailed immunophenotyping of lung disease. IPF increased monocytes and Tregs that are associated with decreased survival. Increased Tregs can also be found in the lung. And then IPF and FHP are really similar in their monocyte profile and different in their cytotoxic T cells. So we are doing now more samples looking at response to anti-inflammatory, expanding our work to e-buses. And we just finished the single cell B cell and T cell receptor sequences, and hope to share it with you soon. So what do I think happened to IPF now? Basically, and again, some of you have already seen this, is basically that we have this organ, which I simplified, and now we have type 1s dying and type 2s trying to respond. But unfortunately, if they have a genetic predisposition or shortening telomeres, they can't do it. And what happens to them is basically some of them die, some of them remain as intermediates, some of them become aberrant basal cells that secrete all these factors and also release dumps and activate fibroblasts and recruit monocytes. And basically, we have this sort of terrible thing that happens in the lung. And this looks very chaotic, but in a sense, it's really reassuring because now we're not speaking about inhibiting fibroblast proliferation. Now we have a detailed map of the kind of interventions we need to go. And I sort of said this thing. So the idea is that IPF is disease of the loss of the respiratory unit more than anything else. We have the features of proximalization of the distal lung, local progenitor failure with the standard differentiation, emergence of aberrant basal cells, which the shift of the archetypes of alveolar macrophages, recruitment of monocyte derived for fibrotic macrophages, and the distinct changes in the. So go back to what we spoke at the beginning, sort of this idea of, can we actually put this into a model? And I think we can. So basically, if you look at the lung, and remember I showed you this, we know what is the primary cell that the lungs exist for, or what's the unit, the AT1 and the AT2. We have the key support cells. And now we can introduce another really important concept in tissue and systems engineering. So when we look at the lung, there's a clear cell that stands out. That's the alveolar type 2. That's basically a factory for surfactant, regulates fluid absorption. It serves as a local progenitor for 81 cells. So it's doing a lot of stuff. And actually, the 82 cells account for most of the mitochondria in the lung. And we also know that mutation in SPC cause familial IPF, and mouse targeting of type 2 cells also increase predisposition fibrosis. And Mauricio Ciccoli, just to show you, this is actually alveolar type 2 cells. I used to think that they just sit there, but no, in response to injury, they actually move to the site of injury. Now think about how much energy this required when you're also doing surfactant, needing to regulate fluid absorption, so a lot of ATP. So clearly these cells are in a lot of stress. And if there is one important principle you want to know, is that the system will most likely fail at its most stress component. This is true in your hospital, in your family, in your car, and in your lungs. So obviously, the type 2 cell is probably the cell most likely to fail, and that's why it's the key cell for potential therapeutic interventions if we can ever fix it. So going back to the model, this is sort of the way we think about tissue homeostasis. We always have a regulated variable, a sensor, a homeostatic signal, and an effector, and then the regulated variable changes. After a change of command is happening, the effector cells go in, effector goes in and fix the regulated variable. If this doesn't work, you have other alternative mechanisms for damage control. How could our cells fit? Basically, very well. So let's assume that the regulated variable is basically the integrity of the gas exchange unit. And then we need to have a sensor. I'm not sure which cell it is. We have an homeostatic signal, and then at least part of the effector is activation of the type 2 cells, which are supposed to proliferate and replace the type 1 cells. If this doesn't happen, and if they become aberrant basilar cells, now you have aberrant repair. And this signal causes both activation of collagen 15, recruitment of collagen 15 endothelial cells, CTHRC1 fibroblasts, and osteopontin macrophages. And then once you get this, you get the stiff matrix. It just keeps getting worse because now you're losing more of your regulated variable. You're accelerating the failure of your effector cell. So I think that this model explained a lot of the features of pulmonary fibrosis, and we could now start inserting even more details like pathways and interaction. Should I finish here, or do I have 5 more minutes? Okay. So should 5 minutes about new drugs in IPF? Okay. So as you see, it's set up, I can do jump here, and then we finish. So that's actually not the question I wanted to ask, but that's okay. My question is, how does it get us to new therapies? And basically, at least one of the ideas is use our data for drug repurposing. Drug repurposing is definitely faster than new drug development, safer and cheaper. And actually, one of the drugs we have working is nintedanib, which is a repurposed drug. It was developed for cancer. So my group was really interested in the connectivity map. Basically, it's a compendium of gene expression profiles of cultured human cells treated with over 40,000 compounds. The MIT, they developed basically gene expression of every compound. And the idea was to accelerate the drug discovery in cancer. And we sort of thought, maybe we use it for pulmonary fibrosis. The problem is that it was developed in vitro, not designed for fibrosis and not for the single cell resolution. So we actually came up with this. This is my pandemic beard, so that's why it's with the beard. That's 4 or 5 years ago. Basically, we use single cell data to generate cell-specific fibrosis signature, and then use the L1000 signatures to predict compounds, and use later PCLS to demonstrate antifibrotic activity. Jasper Flint, who was just a college student, post-college graduated student in the lab, was tasked with developing this. And basically, he developed, this is sort of the structure of the database. The website is already active. If you scan the QR code, Basically, it will ask for your email, and the password is pure IPF, small cups. And basically, you can use it. And what it does is basically it has different features. You could start with a gene you like, you could start with a compound you're interested in, or a cell type. And you have the cell type explorer, signature search, compound explorer, and sort of. And what you will see is basically a heat map depending what you search. So here you can ask, for instance, this is an anti-IPF score. So yellow is increased and is more anti-IPF, purple is pro-IPF. And you could look at different cell types and different drugs, and you can see that nintedanib, for instance, does have an anti-fibrotic effect in myofibroblasts and in macrophages, and maybe in endothelial cells, but actually not in not doesn't do anything in epithelial cells. And other drugs are sort of pirfenidone doesn't do much. Simvastatin rolls out and don't do much. But sarcatinib, actually, which is a drug we followed up, is predicted to have a lot of antifibrotic effect. Here is looking just at the effect of all these drugs on, in this case, collagen expression. And you can see again, sarcatinib has the best effect on collagen expression. And there's a multiple other things. Go on the website and play with it. Unfortunately, this was designed with the thought of a lot of funding coming through. But that funding didn't come through, so it's run basically on Amazon web server with my credit card. So it goes a little bit slow. I also know if people use it because my credit card charges increase. It's my Yale credit card. Don't worry, it's not taking away milk from my kids or something. Anyway, so one of the things that we saw was actually the drug signatures suggest that sarcatinib is a good target for IPF. And then together with Greg Rinaldi's group, we actually submitted an NIH grant that did all the preclinical work. Sarcatinib is a SRC inhibitor developed for Alzheimer and cancer. And we showed all the preclinical work to show that it's probably an antifibrotic drug, and started a trial 3 years after the initial observation. The pandemic sort of slowed it down, but I'm really glad to say that the last patient was dosed a few months ago, and we're now de-identifying the patients, and we'll know some results. So drug repurposing definitely in the drug development. This was done basically with NIH funding and then with the sort of neutral support of AstraZeneca that actually gave us. But there are better ways. The problem with our method that is sort of crude and not computationally sophisticated. So we developed a tool, Jin Dung and my group developed a tool which is called Unagi, which is actually applying AI approaches, so deep neural network framework to study time series and then use this time series data to come up with hypotheses about changes that cells undergo to develop disease. So basically, it does the same clustering that we did before, but it decides on its own what are the cell and how are they related to. And for instance, here, it could show you that, I don't know, the lymphatic endothelium, nothing happens to smooth muscle cells, nothing happened to erythrocytes are sort of splitting into two. These are basically dying. And alveolar fibroblasts are basically splitting to multiple groups as the disease progresses, which fits the sort of the functional characteristic that I mentioned. We can also identify the regulatory networks of each one of these pathways. It's really cool and a lot of fun. Once the software identifies also dynamic markers, which we hope one day will be used to develop biomarkers. But at this stage, what we had is for every one of these tissues that we did the analysis, we also had decellularized protein. So we ran proteomic assessment, and actually, as you see from the heat maps, you can see that all of these markers were validated, which was reassuring. And the Unagi tells you the perturbed pathways and therapies that could reverse them. The difference is that it does it on multiple cells in a very detailed way. And this is sort of one of the prediction. We could actually run different types of prediction. What we really liked this prediction because it did have nintedanib in it. And then what we did is we decided to check the prediction of Unagi using precision cut lung slices. So we picked two things. One is nintedanib, and the other one was nifedipine. Nifedipine is a calcium entry blocker that some of us may be taking if we have blood pressure, very commonly used. And I'll skip this. And basically, we tested this on precision cut lung slices. So the cool thing when you do single cell, you can measure the level of collagen in the therapies, and you can see indeed that collagen 1 is decreased by both nintedanib and nifedipine, and CTHRC1 is decreased, and all the stuff, and you can count the cells. But from my point of view, this is way more important, which is because we're doing, instead of looking just at collagen, we're looking at single cell data. Now I can calculate the distance of the precision cut lung slice from normal and say whether the drug fixed the actual lung, not only reduced collagen. And this is basically the control of fibrosis from control. This is the distance. This is the distance of nifedipine to control, nintedanib from control. And then if you put nintedanib in nifedipine, you theoretically normalize the tissue. We have to be very careful about this. But the other thing that we can look is that we can actually look at the computational prediction of Unagi. How would nifedipine affect the gene expression of specific cells? And what is interesting, and then we measured it in the PCLS, and basically we saw a perfect correlation between the gene expression changes and the gene expression prediction. So I think that sort of AI-based application to dynamic time course data is probably going to be our best way to predict drug repurposing and de-risk drugs for clinical trials. The only problem, we will need to generate the data. And a project like this to do with 40,000 compounds, well, yeah, is beyond anything that anybody would give us. So this is sort of the model of IPF. I think I sort of explained the way we think about it. And hopefully I convince you that at least for the samples we analyze, it is the sort of the failure, the loss of the gas exchange unit and the failure of the compensatory system that describe the disease. Every end of the year, the people who are in the lab, kids, family, significant others show up, and I cook for everybody, and we have a lot of fun. This year we'll do it in a few weeks, and Thanksgiving. And I'm showing this because everything I told you today could not have been done without all these people and many more. And I'll stop here and thank you for listening.
So thank you, Naftali. That was amazing. Have you got any questions from the audience? Peter.
Oh, thank you very much for an excellent talk. A lot of information. Do you think you can isolate these aberrant basophil cells? Because it seems to me you don't know their function.
I think
do you find them in other fibrotic diseases, including other lung fibrotic diseases, but in cirrhosis? I mean, are they linked to fibrotic diseases, or is it a lung type thing?
So I'll start with the first thing. Now that we can make them in PCLS, it's much easier for us to just create a single cell solution and plate them. So we're going to do this because I completely agree. If we cannot modify the function in vitro, we'll never understand what they're doing. The fact that they're sitting there. So we're doing it. And we're starting with a very simple approach. We're just going to chop out P63 or chop out P21 and just see if we're shifting other things. My lab has been working on a completely different direction, which fits to some of the things that actually Gisli is thinking of, which is we are trying to make basal cells into aberrant basophil cells, but just nuking them with things. We're getting to relative similar features, but we never get to the full thing. But we're now trying to do it with type 2 cells. The one commonality, which is very obvious, is when we use LPA, not TGF-beta, we're getting expression of MMP7 and alpha beta 6 into both cell types, which is really interesting, not what I expected. But this is interesting because it suggests that this could be a, how would you call it? A phenomenon, yeah, a cell autonomous phenomenon, something that happens in this. The other side, we do find them in all fibrotic lung diseases. We think we found them also in end-stage COVID, the fibrotic, and we think we found them now in the kidney. Or at least we found cells in the kidney that have epithelial markers and cadherin senescence. And this, we have not found them in the liver yet. But it's a little bit, we just need to download all this data and basically what I call look for our functional orthologs of cell types. So it could be that it's more of a epithelial organ type of phenomenon. I doubt that it would be just lung specific. And we don't, oh, by the way, we don't see them in the gut. But I think the gut, the interesting thing in the gut, actually, when you look at gut strictures, like patients with inflammatory bowel disease, there's no epithelium there. It's really like denuded. It's very interesting.
Great.
The lungs would have the epithelium because everything else is exhausted. What would your prediction be if we targeted that? What would replace the other basaloid cells if it plants to the lungs?
I don't think that that's a failed. I think actually the what I call bady basal cells is probably the last attempt of the lung to repair itself, because I think what happens is the lung learns that it cannot fix the alveolus, but it doesn't know how to build an alveolus without building an airway. So I think that the next step, once we fail for local repair, is that we start to create these weird versions of the airway, which has collagen 15 and basal cells and potentially other things. And, you know, and the only thing that doesn't fit this hypothesis is the fact that we don't find airway fibroblasts in fibrotic lungs. But maybe maybe it's because the way we prepare the tissues, because we're really obsessed with parenchyma, alveolar parenchyma. I think that if you kill the aberrant basal cells, or you probably break some of a feed-forward loop, so you slow down disease, you have to still find a way to improve differentiation and repopulate with the type 1 cell.
So I'm going to disagree with you now.
No problem at all.
So I think we've spoken about this and we disagree where they come from. But one of the things we also see in fibrotic lung, so absolutely loss of type 2 cells, loss of type 1 cells, increase in KRT17, KRT5 negative, aberrant basaloid cells. But you also see a huge increase in normal basal cells in fibrotic lung. So it's not just, so then in normal lung, you only see them sub-epithelial region. But in fibrotic lung, you see a big increase in normal basal cells in fibrotic lung. So I mean, it doesn't make any sense to me that when you're losing AT2 cells, that you've got this big increase in aberrant basaloid cells, as well as a big increase in basal cells. That somehow these AT2 cells are turning into aberrant basal cells before they're trying to turn into AT1 cells. I mean, they could be, and your trajectory analysis is compelling. But intuitively, I don't see it.
No.
But it would be interesting. I mean, and so I would have thought that targeting the epithelium in IPF, there's got to be some way of turning these aberrant basaloid cells into an AT1 cell. If you could do that, then there's a possibility of repair.
I doubt that you could do it, because
think
there's senescence, right?
It's a highly unlikely, but it's
No, but I think actually it's not only basal cells. One is, there's a lot of what I call bady basal cells, because I don't have a good name for them, right? Because they're just different. They're more inflammatory. They have other features. And then, but you have also more ciliated cells. That's a weird thing. Suddenly you have an alveolars, ciliated cells. So, and that's why I say that's the effort to build a new airway. There's no other way I can explain it. It's like that actually what we call fibrosis is lung regeneration without the proper context and structure, right? But I actually think that the alpha v beta 6 inhibition studies will help us, because if the drug works, it may suggest that because there's two cells that express alpha v beta 6 in the lung, which is basal cells and aberrant basal cells. Everything else we said previously, both me and you, is wrong.
Yeah, no.
Right, yeah. So I think if you're inhibiting, it suggests that we're basically pointing to the pathogenic role of these two populations, which I think would be great. Some people are looking at developing CAR Ts for aberrant basal cells. I don't know that will work.
I somehow think killing the average base itself would be bad. But on that note, we're way over time, which is great, which speaks to the fact that this is an amazing talk, and you know, people are stuck the course. And but I think they'll be coming in and removing us soon, and the cheese and wine is going cold.
Okay.
So let's continue the conversations outside. And once again, thank you very much.
Thank you. Thanks for waiting.